Good evening, everyone. How are you guys doing? Welcome back to another stream. I hope you guys are doing well. If you have been following us on our YouTube channel and watching the videos that our other teachers are posting, make sure to subscribe the channel, share it with your friends, and you will definitely be very surprised with the amount of topic that we are coming up in the upcoming weeks as well. I'm hoping that today also you're going to be with me for 15 minutes. We are going to discuss about something very interesting, and that is how to do a monohybrid cross. Monohybrid cross is amongst one of the most sought out question in your 10th standard board. Monohybrid cross and dihybrid cross. These two questions, you'll definitely get dihybrid cross in your paper for sure. Not only because it's one of the important topics for your curriculum, but it's also a little difficult to establish. They might ask you a question, how, establish the phenotypes and genotypes based on dihybrid cross. And it's very difficult if you don't know how to actually establish all the criteria and end up with the proper ratio of the dihybrid cross phenotype and genotype. So that's what we're going to do today. Now, in the upcoming videos, I'll come up with dihybrid cross as well. Today, I want to teach you how to actually do monohybrid cross. But before we go to that, make sure to subscribe our channel. We here at ODA Class love teaching our students in the most innovative way possible. Our application has so many gizmos, it will take you at least 1 week to just go around and see how we actually do the classes. We take free classes also in case you want to give it a try. Download the app. You can find it in Apple Store as well as Play Store for your Android devices. You can even install it on your browsers or on your Mac, on your laptop, as well as iPads. Enjoy the classes for free in case you want to. But if you like our free classes or you just fell in love with our YouTube channel, make sure that you go and enroll for our 1 week course as well. We have a wonderful 1 week course that comes with everything that you can ask for. It comes with amazing amount of content, different subjects, different topics, as well as teachers who are graduated from IITs and NITs. You're not going to find these teachers anywhere near your house most of the time, unless until you're amongst the luckiest one. So why not access that resource and get an experience of how does it feel to be taught by these teachers? We here at ODA Class have been teaching for more than 2-3 years now, and we can definitely surprise you with our method of teaching. Make sure to go ahead and enroll if you want to study with us for 1 week. Not only you're going to get a set of teachers from NITs and IITs, you will also get a specific teacher who will help you out with your any kind of educational queries. These teachers are very experienced in handling students. They can help you with your doubts, with your project work, as well as any kind of school homework that you're stuck with and you don't know how to solve it. Now, if you do find it interesting and you want to join us for 1 week of course, make sure to go to the description of this video. Find the link in the description, click on it, and it will allow you to enroll. You can enroll for just 29 rupees, which is a special offer going on right now because we are starting a new educational year. So go ahead right away, click on this link and enroll yourself into a 1 week course. I hope to look forward to meeting you in one of those 1 week classes. The new batch is about to start from Monday. So this is like the right time. Today and tomorrow is the perfect time to enroll for it so that you don't have to wait for more than one day, which is Sunday. Now, moving forward, let's talk about what we have in store for you today. My name is Sourajit Das. I will be your teacher here. You can call me Jeet sir if you like. That's the easiest way of going around with my name actually. And I'm a molecular biologist. I was in research for a while and then I joined teaching and I love teaching. It's part of my hobby as well as part of my passion too. So I'm going to teach you biology today. As I told, today's topic of discussion is monohybrid cross. One of the most important topic for 10th standard at least is monohybrid cross. By the way, even for 11th and 12th standard, this topic is important. If you pursue biological sciences or let's just say medicine or any other science that includes biology in it in future, genetics is going to be a big part of it. This is basic science now. So let's talk about monohybrid cross. Now, if you notice in the GIF here, you can see there are two parents here, isn't it? One of them is green and round, another one is yellow and round. But for some reason, all the babies are yellow and round. Now the father is quite surprised, like, ये कैसे हो रहा है? How come one of us is green and none of the babies got any of the green trait? All of them are yellow. This is strange. How is the color of the mother getting to all the kids? This is not fair. I'm also the father. So that's how we can understand monohybrid cross as well. So let's get on with it. and understand. Now, before we go in depth with how to establish a monohybrid cross, first of all, let's understand what is a monohybrid cross. Mono basically means one. Hybrid basically means a heterozygous. Okay, I'll come to what heterozygous actually means. So monohybrid cross basically means a cross between two organisms. One has to be the father, one has to be the mother, of course, with different alleles of a single gene. Remember, different alleles of a single gene. If you don't know what are alleles and genes, go check out the other videos that I took in the last week. I've explained alleles and genes and what they actually mean in those videos as well. For now, all you need to know is one single gene can have multiple versions of itself. One version might give you a different trait. Based on that, those versions are called multiple alleles of a single gene. So here we are talking about only one gene itself. For example, let's say one of the gene in our body gives a specific color to our iris, which causes us to have different colors of eye. Now, one version of that gene, let's just say allele, might give you blue color eye, whereas another one might give you brown color eye. So do you see the correlation? The gene is single, eye color, but it can have multiple versions of it. One gives you blue color eye, one gives you brown color eye. So here, that's what we are doing. We are trying to find one single gene, focus on that for the experiment, and make sure that it has different alleles with it. Okay, now moving forward. Now, when you establish a dihybrid cross or monohybrid cross, there are a few simple signs that you have to remember. And during writing all this thing, you cannot be an amateur and write everything. So narrow it down. Use signs that means something. For example, whenever you see somebody writing P when writing down the Punnett square for a monohybrid or dihybrid cross, P usually indicates the parent generation. G, on the other hand, indicates gametes that are going to be produced from the parents. F1 usually means first generation of the progeny or offspring, whereas F2 usually indicates to second generation of the offspring. When the parents commune, they will form the first generation, and then we will take some siblings from the first generation and do another cross to find the second generation. Now, here are the meanings of what it actually going to be throughout the entire discussion. First is the homozygous. If you don't know what homozygous is, this is for you. Homozygous basically means that individual has inherited the same version of that gene. Let's just say that person has only inherited both the alleles, which both of them gives you brown color eye, no other color. So it's a similar material, isn't it? On the other hand, heterozygous or hybrid individual carries different versions of the single gene. Maybe one copy of the brown color gene, whereas one copy of the blue color gene. Just to get a little clearer idea, focus on this one. Here you can understand what is happening. This one here, we have taken a homozygous individual. We have taken another homozygous individual here. So first thing you have to do is choose homozygous parents and perform a cross. Let's just say this is the father and this is the mother in this case. Okay, we have found two parents who has a homozygous characteristics. This parent has a homozygous recessive allele for green color seed. Let's just write it as green. Okay, so green color is here recessive. Remember, it doesn't have so much of expression power when it is combined through next generation. Whereas this individual here has another homozygous, not recessive but dominant. Okay, this person has a dominant allele, which is yellow. So what did we establish so far? Here, the color giving gene has two different versions. One is green, one is yellow. The yellow one is the dominant one, whereas the green one is the recessive one. Now moving forward, once we perform the cross, let's just go ahead. So we are going to perform a cross. Now I know it sounds very interesting performing a cross. Basically, they are just going to copulate and give birth to babies. And only then only their gametes will combine. Now understand how the gametes are going to form. Now we know that both the individuals are homozygous, isn't it? They don't have different versions of the same gene. They have same identical versions of the gene, whichever version it is. Now, if you remember from the law of segregation and law of independent assortment, all of these genes have to be segregated into gametes, irrespective of who it is, best friend or not. they will get randomly segregated everywhere. Now here, if you perform a gamete formation, no matter which gamete comes out of it, everyone will have only that single gene, isn't it? There is no other gene. It's a homozygous. So that's why only one gamete. Same thing applies to the other parent as well. So the mother and father both are giving a gamete. The father gives you the recessive green color. The mother gives you the dominant yellow color, matriarchal society here. And they combine and you get the first generation of babies. Now, when it comes to first generation of the babies, in here, both of them are heterozygous. Why? Both of them contains two different versions of the same gene, not similar version. So here, can you guess what will be the characteristics of this seed? Remember, yellow color was the dominant one and green color was the recessive one. If you know the answer, put it in the comments and let us know. I'll give you a few seconds. You can even pause the video right here and try to figure it out. Now, if you haven't figured it out, let me tell you, both of them are going to be yellow color because the yellow color gene is the dominant one. So the green one is, pathetic. He's not even going to get expressed. The yellow color one will dominate over it. Both the individuals will produce yellow color seed. Now, these two people are actually hybrids. If you remember, they're heterozygous, right? So these are hybrids. That is why we call it as monohybrid cross, because here we are only working with one single gene and two different alleles of the same gene, and they are both hybrids. Now, when you calculate how these hybrids, if they cross again, will give birth to babies, that is what is called monohybrid cross. Here, as we already know, the yellow color gene is the dominant one, whereas the green color one is the recessive one. Keep that in mind. Now we've performed something called as a Punnett square. Punnett square basically allows you to understand how the gametes are distributed here and how they will actually combine. So take a look at this. The male gametes, same, because they are heterozygous, so they will produce two different gametes with two different versions of the same gene. So here you have two different versions of the same gene. The mother and father, both of them are giving the same. Now is the fun part where you combine them in the box. Just combine them exactly how they are. The yellow, green, yellow, green, yellow, yellow, yellow. Once this part is done, comes the second and the last fun part, where you calculate the phenotype and genotype. Now, what is a phenotype or what is a genotype? Phenotype basically means the physical appearance. The organism might have two copies of that gene, yellow and green, but the green one is useless. Yellow one is the one that is getting expressed. So when you look at the seed, it's going to be yellow. You will never know that there is another gene inside which is not able to express, but the yellow one. For example, take a philosophical understanding of this. In majority of the Muslim countries, we call them Muslim countries or Hindu countries because majority of the population is following that religion. There are going to be minority of religion following other people who are not counted in the larger picture. Same thing is happening here. In case of phenotype, you look at the outside version of the organism. Does it produce yellow seed or green seed? You don't go in the genes. Genotype is where you actually go into the genes and understand what combination of genes do they have. Let it be whatever expression it is. So when it comes to phenotype here, these three people, all of them contain one copy of your dominant yellow gene. That means all of them will end up having yellow color seed. That is the reason why you have three right here. This poor guy, he didn't get any of the dominant version of the gene. He got both the recessive one. So he's going to be green. That means yellow is to green is going to be 3:1. That's your phenotype. Now, except the phenotype part, let's just go into the other part, which is the genotype. Let's just remove all of this. If you go into the genotype, this time we are looking into the combination of genes, not what it is expressed eventually. Here, this guy is very unique. This guy is also very unique. He has homozygous green and he has homozygous yellow. But these two people are the hybrids eventually. So what does it come across? 1 for yellow, 2 for the hybrid, and 1 for the green. So you get 1:2:1. Whenever you're concluding after a monohybrid cross in your exam also, always try to write down a conclusion properly that a monohybrid cross of, your hybrids are right here. Both of them contain a different set of genes, will give birth to these individuals. As you can see here also, yellow color, yellow color, yellow color. That's the phenotype, by the way. Look at them. All three of them are yellow. Only one guy is green. That's why 3:1. Whereas genotypically, if we see, this is one combination, this is one combination, and this is two combination right here. That's how you have to conclude it in exam as well. I hope you had fun learning this new trick here. Make sure to watch the YouTube video once again after the streaming if you want to revise what we just discussed. Trust me, you will get it. Give it a try to practice. By the way, talking about practice, we provide practice material in our apps as well. You can actually go and enroll yourself, not only for 1 week of course, you actually might get taught all these things once again in more detail by teachers who have been teaching 10th standard students for 5 to 6 years. So if you want to experience how veteran teachers teach you, make sure to subscribe and go enroll yourself for a 1 week course. It's a very easy process to enroll. All you have to do is go to the link in the description and click on it. You can be the lucky student who might get the entire price of, you have to pay only for 29 rupees. By the way, this 29 rupees can be cancelled out if you take all the classes for 1 week, because when you study with us, not only you learn, you earn as well. How? You get to get coins. These are kind of like cryptocurrencies for our ODA app. We call it ODA coins. 4 of these coins are actually valued for 1 rupee. So you can definitely not only win these coins, you can actually cancel out the 29 rupees that you have paid for 1 week course. And if you like the 1 week course, you can actually even go for a year-long course as well. So what are you waiting for? Make sure that you give us a subscribe for the video, like it, and go enroll yourself immediately. And why only learn when you can earn with it as well? I hope you enjoyed the video today. There are so many amazing videos given by our amazing teachers every single day. You have to subscribe the channel, otherwise you'll miss out on it. So click on the bell icon so that you get a notification every time we're streaming a new episode. I will come up with more of these videos, specifically dihybrid cross, which is going to be a little more complicated than this one. So stay tuned and make sure that you join us again. That's all for today. I hope you get a wonderful weekend ahead. That's where I sign off. Thank you very much for joining in and have a wonderful evening.